Nitrosamine drug substance related impurities. Key points. The impurities are also called as or abbreviated as NDSRIs. These impurities have structural similarity with the API. That's why these are called as nitrosamine drug substance related impurities. This video is for the key points which are repeatedly asked or which are very important to understand, and these points are risk assessment, testing for nitrites in the raw materials, steps in the risk assessment of NDSRI impurities, risk assessment for multiple NDSRIs, NDSRI test as a part of specification, confirmatory testing for NDSRIs, stability testing for NDSRIs, NDSRIs level higher than the acceptable intake levels, and how to set the specification limits for the NDSRI impurities in the APIs and the drug substance. So we know that nitrosamines are formed because of some nitrosating agents and favorable reaction conditions, and the secondary amine groups as the functional groups. The risk assessment for NDSRI are dependent on three key factors: functional groups, amine groups, secondary amines, which are uh, for the nitrosation vulnerable functional groups, nitrosating agents like sodium nitrites that may be present in the raw materials, and the reaction conditions which are favorable like acidic pH and the heat in the process. So mainly functional groups and nitrosating agents are very critical. So testing of nitrites in the raw materials, as we know that nitrites are the nitrosating agents. So uh, there should be a testing for nitrite in the raw materials. Significant impact is there if the nitrites are present. If there is no regulatory limit for the nitrites, the control strategy can be made based on to the experience of the nitrite levels and the NDSRI levels. If the NDSRIs are formed and if you find that the nitrites are present, so critical level should be evaluated. Defining safe nitrite level. There is no specific limit for nitrites. Some molecules may form NDSRIs at some different level of nitrites. So there is no specific limit, but the study or understanding based on the experiments or based on the specific product, we can derive some nitrite levels. Which nitrite level is safe and which will be difficult to deal for the nitrosamine formation or NDSRI formation. A theoretical approach estimates nitrite limits based on the stoichiometric amount required to form NDSRI at AI levels, assuming 100% reaction efficiency, but this requires validation through the practical studies. So practical studies are required to be done. Many of the excipients are there with low nitrite levels, so these can be tried. So now come to the steps in NDSRI risk assessment. First, we have to identify which are the potential sources for nitrosating agents, which may be raw materials, packaging materials, some storage conditions, equipments, and processing parameters. Then vendor qualification. We should ask the supplier for nitrosamine declaration or nitrosamine risk assessment and the nitrite levels of their excipients. Component wise evaluation can be done for the presence of the nitrosating agents, favorable reaction conditions, and the secondary amines. Risk identification and testing. If the risk is identified, then confirmatory testing is required so that we can test and confirm for the probable nitrosamine. Control strategy development. This is based on the risk assessment and confirmatory results. If you have the positive results, there will be a risk and control strategy should be there. If there is no risk, then there the control strategy will not involve the critical steps and you will continue to monitor the product for the NDSRIs. Then by following the structured approach for the risk assessment, it will ensure compliance, product safety and the regulatory acceptance for mitigation of the risk of nitrosamines. How to handle or how to deal with the risk assessment for multiple NDSRIs? When multiple NDSRIs are possible, so this occurs when drug related substances or multiple drugs in the formulation have nitrosation vulnerable groups. You might come across some fixed dose combinations in which some of the APIs have the amine groups. This requires a chemistry based assessment to identify most probable NDSRI as a surrogate for the others. So testing approach, if a probable NDSRI is identified, confirmatory testing is conducted only for that one. If no single surrogate can be determined, all potential NDSRI require confirmatory testing. Control strategy, based on 3 or more batch results, we can define routine skip or no testing is there. So control strategy is dependent onto the results. Establish a total nitrosamine limit, ensuring it does not exceed the most potent NDSRI limit. Impurities less than or equal to 10% of the specification may be excluded from the total impurity calculations. So this is regarding the multiple NDSRIs. NDSRI test as a part of specification. So when we should include NDSRI testing or NDSRI specification into the specification of the product or not. The specification limit for NDSRI is calculated as per the calculation for acceptable intake limit, which is based on the maximum daily dose. The decision include NDSRI test in the specification depending on to the assessment and confirmatory testing results. First, we have to do the risk assessment. If there is no risk, then the NDSRI will not be a part of specification. If confirmatory testing results are consistently equal to or above the 10% of the specification limit, testing may not be required. But if the results are between 10% to 30% of the specification limit, then skip testing can be applied. If results exceeds 30% but remain within specification, routine testing is necessary. So this is dependent onto the market and a specific regulatory body requirement. It is not fixed for all the markets and all the regions. Confirmatory testing for NDSRI. So no risk, no testing is there. Then at risk APIs, if raw material contains some vulnerable amine groups, it is classified as at risk API regardless of whether a nitrosating agent is known to be present or not. The main consideration is for the API with amine group. If it is secondary amine, it is most uh, vulnerable for the NDSRI formation. Then is the risk identification. Some exceptions may be there if stress conditions confirm or stress studies confirm that NDSRI formation is impossible, then API can be considered as no risk. But we don't know on a shelf life period or a long-term storage period, these may be formed. This is product dependent and NDSRI formation sometimes may take place at some different conditions also. Testing requirement. If the risk is identified, confirmatory testing is essential to assess the actual risk and define an appropriate control strategy. Stability testing for NDSRIs. Now, it will be a risk-based decision. Stability testing for NDSRI depends on the likelihood of nitrosation during storage. It is actually related to the research phase. How is the data from the research and development phase for the product or drug substance? Based on that, the risk-based decision can be made. Excipients with trace nitrites. If excipients are present and these excipient could contribute to nitrosation, then stability testing is recommended since safe nitrate levels in the excipients are not well defined. We don't know that what level of the nitrites or nitrosating agents in the raw materials may lead to the formation of NDSRIs. No excipients, lower risk. If no excipients are involved, like in certain parental formulations, there is no further nitrosation risk. Stability testing for NDSRIs may not be required. This approach ensures that NDSRI formation risk during storage are scientifically assessed and controlled. Many of the times, you can study the API, you can understand the API behavior for the NDSRIs, and then you can take a call for the stability testing. Also, stability testing decision is a risk-based decision, and it is a decision based on some scientific studies. NDSRI level above AI. So, what will happen if you come to know that the NDSRI is crossing the acceptable intake level? So, for approved products, notify the regulatory agency which will assess the risk versus benefit and recommend actions. Sometimes, some for some of the products, the regulatory body can accept the product with some level above AI. They can provide you the interim limits, or they can give their own limits, which may be sometime higher for some time till the acceptable intake levels are confirmed based on the toxicity studies or carcinogenic studies. So that time interim limits are provided. For products not yet approved, implement corrective actions to reduce the NDSRI levels below AI. And the actions will include avoiding reagents conditions that trigger nitrosation, identifying and mitigating nitrosation sources, changing the API source if necessary. What will happen if you change the API source? Sometimes route of synthesis may be the reason for some nitrosamine or NDSRI formation. If reduction below AI is not feasible, then establish safety data to justify higher limits. Follow regulatory guidelines for risk assessment. The goal is to have the safe product. Setting specification limits for NDSRI in the APIs. So key considerations are there when the API is in salt form or it is in some uh, complex form. That time you should take the maximum daily dose of the API which is administered in which form. Like if it is administered in salt form, then you cannot take the base form MDD. You should take the total form of the API as a maximum daily dose. Many of the time it happens that people may get confused regarding the label claim and what type of the amount is being added into the formulation. For example, XYZ API, hydrochloride salt, or some different monohydrate form, dihydrate form, or some salt form and hydrate form is equal to XYZ quantity of the actual API or the base. So, in this, don't get confused. Use the whole form which is used in the formulation. So, AI is the specification limit is calculated from the formula AI divided by MDD. So, impact this will result in the lower limits, and it will be always good to have the strict limits. Otherwise, you will calculate by mistake by the acid form or base form. You will have the relaxed limits, and then after application of the product into the uh, regulatory, uh, you will find that the calculation is wrong, and that's why the product is passing. So strict control should be there. This is the information regarding some key points. I have made many videos on nitrosamine and NDSRIs. You can go through those videos, and uh, you can get the information. Thank you for watching the video, and I request you to please do like, share and subscribe to this channel.